All right, good afternoon. I think we're going to go ahead and get started. My name is Yael Mosse, and I'm from the Children's Hospital of Philadelphia, and I have the pleasure of chairing this workshop with Brian Alexander from the Dana-Farber. And we've been charged with having, hopefully, an interactive session, and I'm sure you'll all stay awake because it's absolutely freezing in here, entitled Precision Medicine, Optimal Trial Design, Predictive Biomarkers, and Efficacy. And I hope that throughout the four speakers, you'll hear some different perspectives with myself and Gide really talking about the challenges of the clinical trials, specifically for children with cancer, and then having Brian Alexander and Don Berry really think hard about innovative trial designs in an era of precision medicine and new biomarkers for integration. So I will go ahead and start our session with the first talk today. So to start this session, I thought I would just put out there some of the unique challenges in pediatric cancer, where we're all pretty cognizant that there's a paucity of recurrent somatic mutations in diagnostic tumors that can be drugged, which limits our translational opportunities. Our standard therapies remain largely empiric, although that is certainly changing. When considering an invasive procedure in a child, such as a biopsy, there must be prospect for benefits, so the considerations are different than in adult cancers. There is still a significant lack of access to relapse and metastatic specimens for molecular characterization, and little is known about the relapsed pediatric cancer genome. I found it interesting to go back to what's been published about the NCI match, which really has been the precision medicine trial that's held the promise of genomics and using the discovery efforts to directly impact patients. So the first report came out in May 2016, about a year into the trial, and what this circle plot shows is on the outer edge is initially there were about 700 and see if I can use the pointer. Yeah, 739 cases submitted, of which 645 were able to successfully complete tumor sequencing. This yellow circle is the 56 cases where a mutation matching one of the, at the time, 10 available treatment arms. Then there were 33 patients that met eligibility to be assigned to one of the treatment arms, and only 16 patients who actually then entered a treatment arm. A more recent update provided at a molecular targeted meeting in October of 2017, where the primary difference is that 10 treatment options offered in May of 2016. The time of this update, 30 treatment options. And since the about 2 years lifetime of the adult match trial, 6,000 patients have now been screened. 18% have been found to harbor a mutation that matches one of the 30 treatment arms, and 8 out of 30 treatment arms have now actually reached the minimal patient accrual. So we in pediatric oncology feel that, and I'm sure everyone in the room agrees, that there's an urgent need to test the hypothesis that personalized genomic medicine can substantially improve outcomes for childhood cancer. And I think later on we'll really talk about precision medicine beyond genomics. So in my laboratory, we've been really focused on ALK, and alterations in ALK is a really tractable target, probably one that is unique in that it is an oncogenic vulnerability. There are activating mutations in the kinase domain. And what we have found using a ultra-deep sequencing platform and going back to, sorry about that, sequence 206 diagnostic tumors is that the prevalence of ALK mutations is consistent with what's in the literature. It's about 11%, but using this ultra-deep sequencing platform that was developed by Jolyn Horenza in the laboratory, we're able to identify now subclonal mutations in ALK, really increasing the frequency of ALK mutations at diagnosis. Oops, sorry, clearly having a little difficulty here with this advancing here, okay. And we can use this platform to sequence our patient-derived xenograft models and see a similar frequency of ALK mutations there, where a subset harbor clonal mutations in the ALK oncogene, and a subset, in fact, harbor subclonal ALK mutations, providing us the opportunity to ask the question whether subclonal mutations can be effectively targeted, since that question really hasn't been answered yet. But what we're starting to learn now is that at time of relapse, there's really an enrichment for mutations, not only in the RAS-MAP kinase pathway, as has been talked a lot about at this meeting, but also in ALK, which I think of as being somewhat separate from the RAS-MAP kinase pathway, since we actually don't really know how full-length ALK signals yet. So in a retrospective study of 151 unique neuroblastomas that we did in collaboration with Foundation Medicine, we found that the frequency of ALK mutations at relapse is closer to 20%, and there's a similar trend for mutations in other actionable genes that activate this pathway. We felt that this, along with a lot of other published data that I will not recap today, provided the rationale for performing biopsies at relapse with a prospect for benefit for children. And this provided a lot of the preclinical rationale that led to a CHOP-specific trial that we call the Nepenthe trial that opened about 18 months ago, where we are, for the first time, at least before the pediatric MATCH trial opened, meeting with families and discussing the potential for benefit of a biopsy at relapse, and doing this in collaboration with Foundation Medicine and with our interventional radiologist at CHOP, where a patient with primary or refractory neuroblastoma or time of relapse has a biopsy of a target lesion. For the most part, these are bony lesions. It's rare that these patients have soft tissue lesions. Then this is subjected to next-generation sequencing with the Foundation One platform. There's a turnaround time of about 14 days. And then patients are assigned to one of 3 treatment groups. 2 of the 3 treatment groups are combination strategies based on rigorous preclinical data. And these are meant to capture the patients who have ALK-driven tumors, patients who have tumors with mutations in the RAS-MAP kinase pathway. We find the 2 to actually be pretty mutually exclusive. And for patients who don't have alterations in these pathways but have an intact p53 gene, these patients have an opportunity to be exposed to an MDM2 inhibitor also made by Novartis. What I think is, when we thought about the creation and implementation of this trial, it was important for us that it be reliant on the laboratory-based data and not reliant on a tumor board. And so built into the protocol is a table that, in fact, has specific inclusion and exclusion biomarkers, all of which have been validated in the laboratory, with a lot of sort of, caveats here based on how we define copy number, based on specific residue alterations and what's known about them in the literature. And so there is really no bias or consensus conversation when a patient is assigned to therapy. It is strictly based on a protocol-specified biomarker based on laboratory data. And this is what the algorithm looks like for these 3 strata at this time, where we get a report, and based on the alteration, a patient is assigned to one of 3 therapy arms if they're eligible, and a subset of patients, of course, will not be eligible. And the primary objective of this trial is to assess not only safety, but anti-tumor efficacy as well, since it is biomarker-driven. As I mentioned, 2 of the arms are combination arms. And in parallel, we have the opportunity to perform a number of correlative studies that we hope will help us to understand the clonal and subclonal evolution of neuroblastoma. We're doing some parallel cell-free DNA capturing and monitoring that Chris Bossi will talk about later on in the meeting, as well as generation of PDX models. And what I show you here is the first set of results for the patients who have had biopsies of predominantly bony lesions at time of enrollment onto the study. So these are results for 27 patients. shown here, and the frequency of alterations that we're seeing. And interestingly, in a relatively non-selected group of patients, these are all just patients referred to us at relapse, heavily pretreated, we see that the frequency of ALK alterations, be it point mutations or high-level amplification, is 37%. The frequency of MYC alterations is really consistent with what we know about MYC. Frequency of MAP kinase mutations is 11%. And I'll just highlight here also that we're capturing the tumor mutation burden based on the Foundation platform. And while the mean is about 6 mutations per megabase, there are patients who have mutation burden as high as 37 mutations per megabase, but really quite a range. We also have an opportunity on this clinical trial to consent families to obtaining all of the available archival tissue on these patients. And in collaboration with Jingwei Zhang at St. Jude, we've been able to pilot the sequencing, both whole exome and RNA sequencing of 9 pairs of archival tissue, successive tissue for, well, it's actually 9 patients who have successive archival tissue collected, including the biopsy that made them eligible for our Nepenthe trial. And what I show you here is some of the interesting observations that we're seeing that are allowing us to begin to define the clonal evolution. So this is tissue for patient 002 at day 0. Often this is the diagnostic tissue, an ALK mutation present on tissue obtained at day 292, but then at day 558, the biopsy here shows now acquisition of a TERT promoter mutation, and later on, about 500 days later, we see the acquisition of 2 canonical p53 mutations. Here we see in patients 4 and 6, individuals, this patient here had a TERT promoter mutation, a classic TERT promoter mutation, as well as an SDHC mutation at day 0 and 103. And then at day almost 500, we see the acquisition of an ALK mutation. Who knows if going back to this tissue using our custom ultra-deep sequencing panel, if the ALK mutation was actually present at low level. And similarly, in this patient, no genetic alterations found at diagnosis, but about 500 days later, the presence of an ALK canonical mutation. I'll show you some other interesting results from this initial pilot of these paired samples, where we have a patient with an ALK F1174 mutation, MYC amplification from the primary tumor at day 0, and then day 387, biopsy of a supraclavicular node shows now acquisition of ALK amplification and a new ERB2 mutation. Interestingly, in a different patient, we also see an ERB2 mutation. There are a lot of other interesting mutations as well, but what I show you here is a stick diagram of ERB2 with the canonical mutations that have been described, and the 2 neuroblastoma-specific mutations really reside within those canonical regions. So this trial is ongoing, and it serves as an umbrella trial for us to expand to other combinations and to provide novel therapies as we continue to generate the preclinical data as we learn more about new biomarkers. And in parallel, as many of you know, we've been working very hard to identify more effective single-agent ALK inhibitors beyond crizotinib and ceritinib. And a couple years ago, published this paper showing that this drug, lorlatinib, which is a next-generation Pfizer ALK inhibitor compared to crizotinib, the very first-generation ALK inhibitor also made by Pfizer, is very potent and really is mutation agnostic. So in mutations that are resistant to crizotinib, such as this one and this one in these patient-derived xenograft tumors, we see a pretty impressive single-agent activity of lorlatinib. This is a model that is transiently responsive to crizotinib, and then the tumors become resistant. But lorlatinib appears to be much more potent. And we're very interested in understanding what lorlatinib is teaching us about ALK signaling, and I'll take this opportunity to highlight the work of a talented senior scientist in my laboratory, Dr. Smita Makkar, who's going to be presenting in a plenary session tomorrow morning, a talk entitled Proteomic Analysis Reveals Novel Kinome Profiles in ALK-Driven Neuroblastomas. And very briefly, what I'll share is that we took these patient-derived xenograft models that harbor the 3 hotspot mutations in the ALK kinase domain, treated them with either vehicle crizotinib or lorlatinib, and then subjected them to a chemical proteomics platform that allows us to profile the activation state of the kinome and look at kinome reprogramming. And this is the data for a model that has a F1174 mutation treated with lorlatinib, and then that same model treated with crizotinib. And what is hard to see from where you're sitting, because there's a lot of data here, but these are volcano plots showing the kinases that are significantly downregulated and those that are upregulated. The red are the human kinases, the blue are the murine or the stroma kinases. And what we see, and what Smita will talk about a lot more tomorrow, is that lorlatinib induces profound downregulation of G2M kinases. And we think that this is really integral to its superior activity as an ALK inhibitor in the context of ALK-driven neuroblastomas. So with all of these data, we were compelled to expedite getting this into a clinical trial, and this has now been opened as a phase 1 trial in the NANT, and we activated this trial to activated in September of 2017, so it's only been open for about 6 months. And we have several cohort, one that allows us to do a traditional dose finding. We have a cohort for adults. Many of us here know that adults with neuroblastoma are rare, but they do exist and tend to have a higher frequency of ALK mutations. And then we'll expand and have an arm combining lorlatinib with chemotherapy. We also have a correlative study where, in collaboration with Foundation Medicine, we are able to collect blood throughout therapy and subject it to circulating tumor DNA sequencing and use this to really learn about mechanisms of response and resistance to this therapy. We started off thinking that our preclinical data did not convince us that we needed very high doses to see anti-tumor activity, and so we only planned to study 3 dose levels. But interestingly, we just completed the 3 dose levels and had an opportunity to look at the PK, and it became clear to those of us who really think about the preclinical work and the early phase design of our trials that our levels were subtherapeutic, and that the biology of ALK-driven neuroblastomas is very different than the biology of other diseases driven by ALK fusions like non-small cell lung cancer. And we were able to go back to a model that we treated with a model with a resistant ALK mutation that we treated with lorlatinib at 2 different doses. And in collaboration with Pfizer, we were able to, now that we have a lot of human PK data, especially in the adult trials, recognize that we really needed to probably get to higher doses to really see clinically relevant doses based on the preclinical data. So this is 3 milligrams per kilogram per day. This is 10 milligrams per kilogram per day. So these data have now served as the rationale for us to amend the study to continue to dose escalate, implying, of course, that the patients have tolerated this therapy exceedingly well and there have been no dose-limiting toxicities. And as part of this trial, and I'll just show you this as the last piece of data, we have an opportunity to see what happens when these patients are exposed to an ALK inhibitor when we profile their blood and is subjected to sequencing. And this is just one patient with an ALK mutation, high percent read at time of enrollment, pre-treated with a lot of other therapy, including a lot of other ALK inhibition therapy, and a lot of P53 mutations. But initially she responded. You can see a decrease by about 50% in the burden of ALK detected in the blood. But then just 2 cycles after an initial response, we see clinical progression. We see the dominant mutation is now going back up. But we also see, for the very first time, acquisition of new point mutations in the kinase domain of ALK as a potential mechanism of resistance. So these have been really important early data shedding light on mechanisms of resistance. And so I think I'll just conclude here with just putting forward some of the future considerations as we generate new precision medicine clinical trials, that I think rigorous preclinical modeling is very important to support clinical trials. We need robust responder hypotheses. I think that protocol-defined mutation drug matches is really important to really rigorously test the utility of precision genomic medicine, and then thinking beyond precision genomic medicine, but really consideration of other vulnerabilities, the microenvironment, and moving beyond pure genomics and really thinking about defining personalized approaches when there aren't these more obvious drug therapy matches. So with that, I will conclude. I'm a little over time. Just want to thank the folks in my laboratory and my many collaborators and the patients.